Hello.
Hey, how's it going?
Hey, 1 second. I need to fix sound. Do we basically have the same group, just one more person added?
Yep.
Yeah.
Sounds good.
Excuse me. Well,
There
hey,
you
everyone.
go.
How we doing?
Good.
Good, are you?
Good.
Alive.
Welcome to a new member.
Thank you. I assume that's me.
That is you. Good
So
thing you
who's
showed up. What?
what? It's just us, and then who? I'm sorry, I haven't met. Is Luca? Is that your name? Cool.
Yep.
Hey.
Hello.
Well, this week, at least for me, the material is a bit more confusing and a bit more complicated. I don't know about y'all.
Yeah.
I actually had an easier time this week than last week, so
I agree with
It's kind
that.
of repetitive. I feel like once you kind of understood the hang of it, it didn't. I don't know
mhm.
if that makes sense.
Hmm.
The only thing that confuses
Yeah.
me really badly are the bar bodies. I don't know why I changed.
Yeah.
Yeah. Okay. Well, we can bust this out real quick. Sorry, I'm just finishing up a quesadilla.
I just finished a burrito, so
Oh,
I'm
nice.
sitting at Costa Vida, so
Alex, did you not get the memo that it was like Mexican Thursday or
I
something?
guess
Like,
not. I guess not.
well, all right. Well, so looking at number one,
okay.
I don't even know like if I did this right or anything. Um, so like the Angelman syndrome and stuff. So like if they're heterozygous, they are a carrier, but they're not affected. And then if they're homozygous, recessive, they're affected.
Mhm.
mhm.
Okay. And so like if you're affected, you basically only get your genes from mom.
Right.
Um. So how I did that is I just did like the Punnett square, just did a regular cross for that, and
Yeah.
because it says it silences the paternal alleles, whichever one that the dad gave to each of the progeny, I just crossed out, and that's how you got the phenotype for it.
Yeah, I did the same thing. I just made like the dad's allele. I just put it in a different color, and basically you can ignore it because it's silenced, right?
Mm-hmm.
Mhm.
Okay.
So for part A, you get 50-50.
Yeah.
For the phenotypes.
Yeah.
Uh-huh.
Yep.
Okay.
50-50. Huh.
Yeah.
For the phenotypes, because
Yeah.
you only are looking at the mom's allele.
So like, but it's did I set it up wrong? Because it's a heterozygous cross with a heterozygous. I thought that was a 1-to-1 ratio.
So that's what it is, like genotype wise. But since you
Uh-huh.
or the dads, so like one of the heterozygous ones would be like positive for the disease.
Hmm.
You would just kind of look at the mom's recessive trait, right?
Okay.
Mhm.
Yeah.
Can you guys see my screen with the thingy? I don't know if I did it right. Is that what you
No, it's showing up. Yeah.
Yeah.
Okay.
I'm just on
Okay.
my phone, so it's tiny.
Interesting. Okay. I guess that makes more sense. And yeah, I really didn't do any of those right this week. I'll have to go back and study those.
Um.
I want to take the quiz. All right. So then,
For B, I got 100% disease.
Yep.
Okay.
Yeah, so we basically did the same thing for that.
Yeah.
okay. All right.
And then
And so then looking at C and D, I didn't think that the woman that has the triple X syndrome, I didn't think she would have any bar bodies.
That's
But-
what I heard.
Mm-mm.
Yeah.
Okay.
Okay.
Yeah, she wouldn't have any because the XICs are what are counted. That's how they know to inactivate one or the other.
Okay.
Right. So, did we all get that one?
Mm-hmm.
Yeah.
That's what I got. Okay. And then D threw me for a trip. I didn't know how to start on that one.
Yeah,
I went with one bar body, but
I put 2 because I went back in the notes and read exactly what the T6 gene did. And what I got basically is the T6 gene cancels out the XIST. If you don't have a T6, then the XIST will deactivate both of them.
Yeah. So what I understood from the reading is the X. I don't know. I just call it schist. I don't know if that's bad, but I think what I understood, its role is to deactivate, and it only doesn't deactivate the ones that have the T6 functional.
Mhm.
So we would have 2 at the end, 2 bar bodies.
oh, okay.
Yeah.
Okay.
Does that make sense?
Yeah.
Yeah, that makes a bit more sense.
A bit more.
I think I just need to reread the notes a bit just to like fully like make like a little flow chart or something.
Yeah.
Yeah.
'Cause I just forget what each one means.
'Cause
Like right after I get the problem done, it's just out of my head.
I think like the first one, the X whatever, the schist one, it makes like an RNA, and then it destroys it makes the bar body, and the other one just makes a complementary RNA. So instead of destroying, they just get together. But if you don't have like a shield one, you're gonna be deactivated.
Yeah, so the TSIX or whatever you said, like stops the other ones from deactivating all the chromosomes.
Mhm.
Okay.
Okay.
So only one would be deactivated normally if both were functioning normal.
Okay. All right. Well, if we're good there, I guess we'll move on to number 2 then. So, what did you guys put down for an example of a holoandric gene?
ESRI. Y. Sorry.
Yeah.
What was it?
The SRY.
I1. Okay, yeah. I remember that.
Okay.
And then for a gene found on both X and Y chromosomes, I put MIC2.
Mm-hmm.
Uh-huh.
Is this pseudo-autosomal, something like that?
I don't remember what it actually stood for. Let me go back.
Yeah. I think
thought
it was.
Because it
'Cause
like
if they're both
because what I think it's pseudo-autosomal is like because it's present on both X chromosomes, it can trick you when you're doing the Punnett square, so you can think that it's an autosomal disease, but it's actually on a sex chromosome.
Yeah.
But because it's in both, you can't really tell if there's not really a different pattern from an autosomal disease.
Okay.
Did it make sense? Did it?
Yeah.
Did.
So we back to A and kind of explain what exactly a holoandric gene was.
It's a gene that's found on the Y chromosome. I'm pretty sure that's it.
Mm-hmm.
Is that what it is? Okay.
I'm gonna write that down again. Well, sweet.
Okay.
So the dwarf questions, like the next one on C, and even in the reading, I'm still working on understanding those.
For those, I found it was really helpful to do something like this. And I don't know if you can see, but like I changed it to a simpler thing. So I did 2 N's instead, and that was a lot easier for me to keep track of.
Instead of like all the letters. I like that idea.
Yeah. Okay.
Yeah.
So you got 3 and 1?
Yeah.
I must have done something wrong then.
I don't
So
know.
we do this
I
one the
did.
same as it's the same as the,
The Angelman, right?
yeah.
But it's just like it makes more confusing because instead of 1 letter per allele is like 4 letters, like 3 and a number, I guess.
Mm-hmm.
So-
I think that's probably my problem is I'm looking at that big giant freaking chunk of letters and just like, where do I start?
Yeah.
Can you help me understand what I did wrong right here? Because I feel like, can you see the screen?
Yeah.
Mhm.
Because I put right here that they're both heterozygous. You know, one has the negative, minus, whatever.
Mhm.
And I did the Punnett square and deactivated the lighter color right here, the ones that were from the mom, right?
Yeah.
'Cause and so I thought it was 50-50, right? Because these 2 are dwarf and these 2 are normal.
I didn't know that the moms were not effective. Or where did you get that information?
I think it was from, right, let me check.
It was in the PowerPoint, I think, in the example that they
I
gave.
think it's in the notes too. Where
Yeah, it says-
the
Yeah.
allele from the mother has been inactivated.
Yeah, so
Oh.
it's under genomic imprinting, and it says, right, in the case of this weird allele, the maternal allele is silenced.
Hmm.
Okay, I didn't remember that. Thank you for catching that.
So your work is
Okay.
right. It was just you had forgot like one little step.
Thanks.
Yeah.
So it is 50-50 because that's what I just got. Well, I don't know. But I just got 50-50.
Okay. Yeah, now I got it. Okay.
Sweet.
Thanks for double checking that.
No worries.
All right. And then 2D, I said sex influenced.
Yep.
Yeah.
Sweet. I got one basic thing down this week.
Great.
So,
I don't know why this week was so difficult for anyway. Okay, so then E. Oh, that's not right. Whoops, that was
I put
stupid.
genomic imprinting for the first one.
And dosage compensation in a second.
Yeah.
Mm. Yeah, that makes sense.
Okay.
Do we need to do any more explaining?
No,
How do we explain? I think it's in the notes.
Yeah.
yeah,
There's not much.
I put those ones backwards. Oops.
Because in the notes that under epigenetic inheritance, there's only 2 types, and one says the dosage one occurs during early embryonic development, and the other one says during gamete formation. So I think we're right.
Okay.
Yeah. Okay, cool.
Okay, and then F. So that one for F, the double-stranded RNA, that goes back to one of the X body things.
Mm-hmm.
Which one?
Yeah.
Was it the XIS?
Yeah, it was XIST and the T6.
Yep.
That's how they cancel each other out, is they make that double-stranded RNA, and then the cell destroys that double-stranded RNA.
Okay. Cool, sweet.
All right.
Okay. Reciprocal crosses, identical results.
For G, correct me if I'm wrong, but it was autosomal chromosome and X chromosome.
I put
Sorry.
autosomal genes, but I don't know if it matters.
Oh, I just wrote autosomal.
Yeah.
I guess I did that at the beginning too, but then I saw alleles, so I thought maybe it was chromosome.
Because it's like allele it's from a gene is in the chromosome, so we're all right, I think.
Yeah.
Yeah.
Cool. Alright.
Excellent.
Number 3.
Sorry, I'm rereading what I put on here. I
No, you're
did
fine.
this on like Tuesday, and I was
You're
at
good.
work.
Yeah.
I did it at work. I think that's why I should not do it more at work.
Okay.
Bad idea.
Yeah.
Okay. Father. Well, that's a fun word. Next link, recessive.
Do you want to try to read the name of the disease?
Transcarbamylase.
Yeah.
Nicely done.
So the genotype of the woman's father. The woman's
So it's
father.
X recessive.
And then a Y.
Yeah.
Right?
I put the lowercase d as the
Mhm.
affected thing, so it's XDY.
Okay.
Yeah.
I just put a little plus sign.
Okay.
And then
And like since the woman is not affected, right?
yeah.
Her mother must be a homozygous dominant, right?
Yeah.
Mhm.
Okay.
Mm-hmm.
Yeah, that makes sense. Okay, there we go. Woman. Genotype of woman could produce a daughter.
So for
Yeah, I
the
guess.
second one, to figure that out, you have to cross her mom and dad and see like the different opportunities for her to have the trait, and then you can cross that with a guy that's not affected.
Mm-hmm. Yep, that makes more sense.
So
Okay.
you guys get 1/4.
Yeah.
Where? Like letter B?
Yes.
So if the woman has children with a man who does not have, I did not get just as
Well-
amazing.
If you cross the mom and the dad, the only genotype that the woman can have is a heterozygous. So
Oh yeah, no, you're right. You're right. I had highlighted the sons in blue and the daughters in pink, and I just looked at the blue and said, "Oh, 50%," but it's 25. You're right.
Okay.
Okay.
sounds good.
The genotype of a male with whom the woman could produce a daughter with OTD. What was I thinking when I did this?
He would have to have OTD, wouldn't he?
Yeah.
Yep.
Yep. Okay, well that was easy. Consider the cross described in part C. What are the expected genotypes and phenotypes of the offspring? Well, if
Oh, wait a minute. Just a second. Sorry. I remember why I put 50% on the thing, because it's a 25% of chance of a like son or daughter be born, but it's 50% of chance of a son being born with the disease because it's half, right?
Mm,
Don't you multiply the percent of sons, because it's 50-50 for sons and daughters, and then you multiply that percentage by the chance of having the disease at all, or am I wrong?
I didn't get 50-50 for them having the disease.
Because
I think it's just the Punnett squared percentages, what you get from what you cross.
is it just that 25? Okay,
I think so.
mine. Sorry, where were we? D.
Yeah.
Yeah.
Let's see. So she's supposed to keep the same guy, right? So he has to have it.
He doesn't have it for B.
Right. Are we talking about, like, we're on D though, right?
Yeah.
Yeah.
Okay.
Oh, I thought we backtracked to B. Okay, that's fine.
You're fine. Just throwing me curveballs now.
Sorry.
So the expected genotypes and phenotypes of the offspring. So
You just need to
yeah,
put down what you put for what you got for them.
Mhm.
Is this one 50% then? Because the son and
Yeah.
daughter? Okay.
Yeah. So all the daughters should have it, right? No.
No,
They
just
should
one.
have it.
Yeah.
Wait, just one daughter, right?
I'm confused. I got like a fourth, like for heterozygous, a fourth for homozygous, recessive. Like I got. Sorry, I'll just show the screen. It's easier that way.
Yeah.
Mhm.
Can you see it?
Is
Yeah.
the third Punnett square, right?
Yeah.
I figured out like I can like show everybody like one specific screen, but I don't know how to turn that back off.
So when you click on screen share on the corner, there's like a little tab will pop up on your screen and it says stop sharing whenever you can click it whenever you don't want to share anymore.
Oh, okay.
So if you go to the Hangouts, there's a green screen share thing. And then
Gotcha.
we just have to click on your picture to see what your screen is.
Oh, gotcha. Okay. Sweet. I've never used this before.
Me neither. I had to figure it out.
Cool. Props
Can
to
I-
you for figuring it
Can I
out.
show you? Can I show you so you guys can help me
Yeah.
understand too? Because I'm really confused.
Yeah.
So this is the right genotypes for the parents, right? He is affected right here, right?
Mhm.
Mm-hmm.
Yeah.
And then the mother is heterozygous.
Yeah.
So
Mhm.
this daughter would be unaffected. This son would be unaffected, but these two will be affected, right?
Yeah.
Yeah.
So it is 50%.
Yeah.
Okay. Sorry, just wanted to make sure.
Cool.
Oh, okay. I guess I separated it by their sex, but it just is offspring, so
That's fine.
I don't know. I guess like you could bunch them together.
Mm-hmm. All right. Well, if we're good there, we'll move on to 4.
Mhm.
The beard and goats. I remember this one. I texted my mom about this. We used to have goats.
Okay.
How fun.
Okay. So suppose a cross produces a 3-beard, no beard phenotype ratio among male offspring. What are the genotypes and phenotypes of the parents?
Did you get double heterozygous for the parents?
Yeah, but the
Yeah.
female wouldn't be bearded.
Oh, yeah, because it's dominant. Okay, yeah.
Yeah.
So yeah, the phenotype male beard, female no beard.
Yeah.
Mm-hmm. Okay.
Because for the mother to be bearded, she would have to
Be
have
recessive.
both of the alleles.
Yeah.
Mhm.
For B, isn't it the same thing?
Yep. Because it's inverted.
No, offspring is basically just the opposite of what that question was asking.
Okay.
And then a cross between a male with a beard and a female without a beard produces a 1-1.
I put heterozygote female non-bearded and then a or homozygote bearded male.
Yeah, I got the same thing.
Okay.
How do we show on a video that how we did it? Just show the Punnett square? Because she said she would like dock points if we don't explain. I don't.
Yeah.
I did
I
it this.
Let me see. Hold on. Hold it right there. Everybody sees her screen now, right?
Yeah.
Yeah, you just click on her little picture and you can see.
Sweet. Yeah, that makes sense.
Okay.
Sweet. Okay. And then D is both producing one. Okay. Yep. Simple. Good. All right. We'll go on down to 5 then. Man, this one was a long.
Did we talk about D?
No, excuse me. I did. I thought about it. My bad. Okay. So male and female, both which black beards, do the same thing with a 1-to-1. So
What are the genotypes?
she has to be.
I feel like for the male to lack a beard, he must be homozygous recessive, right? The lowercase a. And for the mother, for them to produce at least one with a beard, she needs to have the dominant allele.
Yeah.
So
She's not
she's
gonna
Yeah.
be bearded, but they're gonna produce one bearded, right?
Mhm. Yep.
Do you have to differentiate like what genotype corresponds to which gender, or can you just say like
I think it's specific to like which one has the female, because if the female had the recessive traits, she would have a beard. I don't know if it matters, but I don't know. Because I have the
Yeah.
female as being the heterozygous non-bearded, and then the male also being non-bearded, but it's the
It's like if the female has both the capital letters alleles, then she'll be bearded. But other than that, she'll never be.
yeah.
Mm-hmm.
It's like dominant for the male, but recessive for the female.
yeah, okay. So
this is
for C, the heterozygote is the female with no beard, and then no beard for the male.
For C?
Yeah.
For C, the male has a beard, right?
mm-hmm.
I think we're at D right now.
Yeah, we were talking about D.
Oh, we were? Okay.
Yeah.
Sorry, can you repeat that again? I was not on the same page. So heterozygote is the male, and then the
You're talking about D now?
homozygous recessive for D is the female.
So in D, I got the male is what I think you said, homozygous recessive, and the female is heterozygous.
Oh, okay. Is it the same for C? Or flipped?
No. For C,
Yeah.
I'll just show you. I'll just show you my screen. Okay. Can you see it?
Yes. Okay. So,
And for D, I just explained because I didn't know if we needed to do another Punnett square.
so I didn't get the same thing for C.
What did you get?
I got the same answer as D.
Let me see.
Yeah, sorry, I got mixed up because the male and female. Okay. We're good. Sorry, we can move on now.
Okay.
Oh, you're fine.
That's why we do these. Sweet. And stoppers. Okay. Okay, so 5. Oh, goodness. Cats. I hate cats. So with these, it matters about. So obviously, big B is dominance of little b, basic, but then these ones are only passed on through the X, right?
Mm-hmm.
Yeah.
Okay, I want to make sure I got that correct.
And it's kind of like a blend, right? Like if they're both like recessive, it's black. If it's both dominant's black, both recessive is orange. And if it's a mix, which would only happen in females, it's the calico or tortoise shell.
So it's like, oh, okay, yeah. It's basically like a sex-linked kind of stuff we talked about last week.
Mm-hmm.
Yeah.
Okay, wow.
A male can be both, though, if he has more than one X chromosome, right?
Yeah.
Going to be kind of.
Like with that weird syndrome, Turner syndrome. What's the name of it?
Yeah. Where he's XXY, right?
Mm-hmm.
Yeah. That'd be weird.
Can it happen with? Wait, no, it's not Turner syndrome. I don't remember the name of it, but anyway.
It's one of those, but we know what you're talking about. Like they're the double X and then a Y. I thought it was Turner's, but maybe not. Never mind.
Turner's is 3 X's, I think.
Okay.
It's Klinefelter, something like that. XXY.
Yeah, I think you're right.
Yeah.
All right. An orange male cat, orange female.
So for the A, I got 2/4 orange, 1/4 black, and 1/4 tortoise shell.
Mm-hmm.
Yeah.
Yep.
Do we need to differentiate them by their sex, though?
Okay.
Because it's sex-linked.
One orange female, one orange male, one black male, and then one tortoiseshell female.
Yeah.
This is, I don't know. I like coloring this stuff on the document. I don't know if you guys can see it.
Yeah.
Cool.
This was supposed to be orange, but it's not.
I think you should probably include, though, the gender in the phenotypes, right?
That's true, I guess.
Just to be thorough.
Yeah. I mean, it's obvious by your Punnett square, but maybe she'll knock you points or something there.
Well, just in case, I'll put it. Thank you.
Mm-hmm. All right. So this question concerns exactly what we were talking about before. Are very rare. What is the genotype of a male tortoiseshell cat? So he has to have that double X and then the Y, and then he has to be heterozygous for both. Heterozygous.
Well, I didn't think of Klinefelter, so I put it was an impossibility because I don't know about Klinefelter syndrome in cats. I don't know if that's a thing. So if you think in a normal situation it would never happen, but I don't know if there's like, I don't know.
I'm sure there's some genetic mutations in animals as well. So
yeah, that one made me think. This one time we were out hunting and somebody took down what we thought was a white-tailed deer, and like we had it on the rangefinder and its tail was white. And when we pulled up on it, it was actually a mule deer, but it had a white tail, which like really isn't supposed to happen at all.
Yep.
It was really weird. The game rangers just like said, "Oh, you're fine," but that question made me think about that, and I was like, "Oh, that one's probably possible." And there was a real life example I had been through with that.
Maybe it was a mix, whitetail and mule.
See, that would be so weird. Like even if like a whitetail and a mule, like one would have. I obviously don't know venison genetics, but I didn't think like they would mix like it wouldn't. I didn't think it'd be like co-dominance or something like that. Anyways, Stop confusing me. I understood that question until you said something. All right. So fruit fly mutation produces scalloped, irregular margins on the wings.
Okay.
Scalloped phenotype is an X-linked recessive mute. Indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done.
Reciprocal cross is when you switch, like, for example, if the dad is recessive and the mother will be all dominant, and then you swap. Is that what it is?
Mm-hmm, yeah.
Okay.
And then if there's a difference, what was it? If there's a discrepancy between the two,
I think if there is a discrepancy, it would be X-linked, and if there isn't, is autosome or something like that.
yeah, that sounds right. I was trying to remember off the top of my head, but that sounds right.
Yeah.
That one, I felt like the question was kind of like unneeded. I don't know. It was kind of simple. I guess it helped you to understand like how to do a cross.
The
Yeah.
reciprocal crosses. Well, all right. And then assume that, and then the question for D, it pretty much told you it was an autosomal recessive kind of.
Sorry, did we get the percentages of the C? I didn't see it.
No. Sometimes we don't say them.
Is it? I didn't know if we were supposed to do it or indicate genotypes and phenotypes. So I got, for one, I got 100% normal, and the other one was half-half. Is that correct?
Yeah.
Okay.
So that means it's X-linked, right?
Right.
Because if it was autosomal, it wouldn't change at all because it's not only X chromosome.
Okay. Sweet.
Sorry for interrupting.
Oh, you're fine.
I'm glad we went over that.
So we're just supposed to do the progeny for the reciprocal crosses for this one then. It's basically the same thing as C.
Yeah.
But this one, because the disease is X-linked, and nothing changes from one cross to the other, right?
Yeah.
4 heterozygotes for each cross I got.
So you do the same cross, but nothing changes.
Yeah.
Okay. Cool. All right. Well, my phone's gonna die, so.
Yeah, I gotta.
All right.
Y'all. Thanks for the video. See y'all
See you
later.
guys next week.
Do you need help posting it? Because there's extra instructions this week.
What were the extra instructions? I didn't see extra instructions.
Yeah, she had this like, yeah, 2 different places.
Use different places to post it. Anyways.
Yeah.
Okay. Well.
I still have like one like, remember that I was like stuck on that 3B. Now I came back to my previous theory because it's asking. Sorry if it's bothering, but it says, what is the chance that the first child will be a son with the OTD? So there's from the sons, half of the sons can be OTD. Isn't that 50%?
Well, I can show you the cross I did.
Because I know that like overall the Punnett square between males and females, that would be 25%. Because it will be just one son that can have it. But it's asking specifically just like one son with the OTD. And it's, I thought that was like if the probability of having a son is half and the probability of having with disease is half of the half, it's just, I don't know. Maybe you're right. Maybe I'm just overthinking.
So can we go back to the phenotypes of the parents? So the female is a heterozygote, right?
Yeah, she's,
Okay, and then the male is dominant.
Yeah.
Okay, so in my cross, I got 2 females. One female was homozygous dominant and one was heterozygous. And then I got 1 male that was dominant and 1 male that was recessive. and the one that was recessive would have the disorder. So there's 1/4 chance.
I got that same Punnett square, but my thought was just like, if it's asking only about the son, you would discard the 2 daughters from the probability. Does that make sense? Because like,
yeah, I see what you mean.
What's the chance of having a son with the disease if you can only have like those 2 genotypes for the son? Half of those genotypes would be the diseased ones. That's what I thought. Maybe I'm just overthinking.
I think, I don't know, I still stand by mine because it says
that's fine.
I mean, like, we can both try it out, and I'm sure she'll like correct us if we're wrong, but it says what's the chance that their first child and then will be a son and with OTD, so I guess that's how I took it. But.
Okay, well, hopefully she gives us like good credit for the video because we debated, you know.
Yeah.
Okay. Well, so I zoned out on both of you. I'm sorry. I was reading the new about the posting the video twice. But to me, it would be you take the probability of them having a son, so like 1/2, times the probability of an offspring having OTD, and then that would give you the chance of having a first child male with OTD.
So it's half times 1/4 with 1/8.
1/8.
Yeah, so they have a 1 in 8 chance of having a kid, having a son with OTD.
Maybe we're both wrong. Maybe he's right. I can't, because I remember doing these multiplications of like different probabilities, you know.
Mm-hmm.
Well, they can only.
The thing is, I think we would multiply the way you were saying if it was like an autosomal disease.
Right.
But this one is specifically the X linked. So I don't know. Maybe it's good that we were talking about this. Maybe she'll give us credit.
We'll see what she says. I mean, I'm sure we can like look it up and spend some time trying to find it later. But I know, at least for me, that's how I did it.
That's true.
Looking at it, but I don't know.
Okay. Well, thank you for waiting and double checking. Sorry that I decided to bring it up last minute again.
Hey, you're fine. That's the point of the video. Anyways, it looks like it looks pretty simple instructions for posting the video, so I'll probably post it tonight, if not early tomorrow morning. But probably I'll just do it now.
Okay.
Thank you so much. I appreciate that.
Thank you for having me in the group.
Yeah, Luca. Y'all have a great evening.
You too. Bye-bye.